When it comes to the word evolution, most people think it's all about fossils and things that happened millions and millions of years ago. However, towards the end of our last discussion, I explicitly told you that nothing can be further from the truth, and there are several ways in which evolution impinges upon our contemporary lives. So, that is the topic of today's discussion, evolution through the lens of application, particularly modern day applications. Now, perhaps the biggest application of evolution is in terms of agriculture and animal husbandry. As we already saw in our last discussion, when humans developed agriculture, when we started domesticating animals, that completely changed the way we lived and that further shaped our own evolution. However, that process of getting hold of animals and other plants and making them work for us from an economic perspective, that is something that has not really ended. In fact, it's, if anything, right now, given that the population of the world is increasing and given that we are kind of hitting some kind of barrier in terms of increasing our agricultural and livestock productions, research in this particular area, breeding animals, breeding plants to make them more economically viable, research in this area is absolutely booming. So, just to give you some example, here are just a few of the national level plant breeding and animal breeding institutes of India alone. And these are national level, which means that at the level of each state, you have many other such institutes. Many universities also work on this topic, and this is just India. You have such things happening all over the world. So, this is just to tell you the scale at which people are using principles from evolutionary biology, breeding and other stuffs in order to improve our crops and animals. However, this is not what I am going to talk about today. This is a very specialized subject. What I am going to talk about in a little bit more detail is about evolution and health. How exactly evolution has ended up affecting the practice of medicine in a rather big way. And there are 2 or 3 major topics over here, but the first topic that we are going to take up is in the context of antimicrobial resistance. Now, what exactly is that? So, antimicrobial resistance refers to the ability of microbes to resist the chemicals that can otherwise kill them, kill the bugs. So, depending on which kind of organism we are talking about, antimicrobials are of different types, and therefore, the corresponding agents resistance is of different types. So, depending on the organism, so for example, if it's a bacteria, then the kind of drug that we use to kill them, they are all called antibiotics, and therefore, you have antibiotic resistance. Similarly, for viruses, you have antivirals and antiviral resistance, parasites, antiparasitic, fungi, antifungal. Now, very often, the reason I am making this distinction to you is very often when we say antibiotic, we tend to think of all microbes, but that is not really true. Antibiotics are only for bacteria. You have all these other classes of organisms which require very different kinds of chemicals to kill them. But together, these chemicals constitute antimicrobials, and the resistance against them is antimicrobial resistance. Now, in 2021 alone, there were about 1.14 million deaths which were directly attributable to antimicrobial resistance, and it is estimated that by 2050 this number is going to be about 1.9 million annual deaths. That is a huge number, okay? And this is only places where things are direct. Okay. If you look at the indirect effects, they are obviously much larger, but more worrisome in this context is the prediction about how these deaths are distributed globally. And this is the predicted death rate per 100,000 per 1 lakh attributable to AMR for all ages in 2050, and essentially this is the color code, and you can see the deeper the color, closer to red, the worse it is. The highest category is this one, and this is India. So, what is predicted is that barring this one place in South America, India is probably going to have one of the highest rates of death due to antimicrobial resistance. And right now, it's a absolute living nightmare for most hospitals in this country. If you tend to go to a hospital and you tend to catch a bug which is antimicrobial resistance, then getting rid of that bug can sometimes be extremely difficult. Now, why exactly do we have this problem? Now, in this context, whatever I am going to discuss, I am going to focus primarily on antibiotic resistance because that is what has been best studied. However, the major things that I am going to talk about, they are applicable across all the classes of drugs that we talked about. Now, many antibiotics, we have to realize, are natural chemicals. They were isolated from bacteria, and they have been evolved by bacteria essentially to fight other bacteria, right. So, because of that, there is always some level of antibiotics in the environment, which means that even without us human beings doing anything, there is always a baseline selection that is happening amongst the microbes to fight each other, okay, and there is a baseline selection for increased antibiotic resistance. Now, of course, because of this and because of the fact that the bacteria have been around for millions if not billions of years, there are many ways in which a bacteria can end up evolving antibiotic resistance. For example, for any antibiotic to work, it has to be able to go through the membrane. So, many bacteria can end up doing all kinds of modifications at the membrane level such that the uptake of the antibiotic itself is prevented. Similarly, there are all kinds of pumps available on the membrane which actively expel whatever antibiotic has been taken. And when I say actively, I mean that in exchange for energy. So, this is the energy consuming process. And these efflux pumps, sometimes they are very specific, that is they will only work for one kind of drug, but many a times they are pretty broad based, which means that one efflux pump can actually throw out many kinds of antibiotics. Then, of course, bacteria have evolved different kinds of enzymes that can degrade antibiotic molecules, and sometimes what happens is that each antibiotic, when it goes inside the bacteria, it typically ends up having some kind of a target site. So, some work on, let us say, topoisomerase, some work on the gyrase molecule, some work on the ribosomes and so on. So, what the bacteria can do is it can modify the site at which the antibiotic will go and bind, and thereby it can prevent the drug from binding, essentially making the drug ineffective. Now, very interestingly, some of these mechanisms are very specific. So, for example, target site modification is a very specific thing which typically works only for antibiotics which will bind there. But some of the other mechanisms, for example, this efflux pump mechanism that I talked about, those are generic mechanisms, which means that they are not only important in the context of antibiotics, they can be used by the bacteria to fight other chemicals, for example, heavy metals, etc., which are entering the body of the bacteria or inside the cell of the bacteria. Now, this particular piece of information is actually very crucial, as we are going to see in the next slide. Now, what are the selection pressures for antibiotic resistance? Now, obviously, the biggest selection pressure is an increase in the level of antibiotics in the environment that has happened over the last 4 or 5 decades. Why has that happened? One major reason is climate change. Now, we know that because of climate change, in general, there has been a warming of the environment all over the world, and because of that, the number of bacteria which are growing has actually gone up in many places. Why so? Because we know that many bacteria, almost all bacteria will have an optimal temperature range in which they can grow, and when the world is slightly colder, then that optimal range is typically not reached. But because of climate change, because there has been warming, now many bacteria are finding that the climate is much more suitable for their growth, and they are, as this study points out, it is increasing the number of taxa of bacteria which are growing. Secondly, obviously human beings have been using and abusing antibiotics for the last 45-50 years at an astonishing rate. So, nowadays if you go to any doctor, okay, not any doctor, but many doctors, they end up prescribing antibiotic without really thinking through whether that antibiotic is really needed or whether the patient will be able to take care of the infection on their own or not. Can't blame the doctors entirely, because if a patient is suffering, they go to a doctor and the doctor tells them that, look, you just fight it through, because antibiotic resistance is increasing, the patient will probably go to a different doctor. So, many times because of those kind of social reasons as well, the doctors are forced to prescribe antibiotics. However, the much bigger issue in the context of antibiotics is in terms of livestock. So, we know that our livestock, the cows, the buffaloes, the goats, the sheep, etc., most of them have been bred for higher productivity, and many of these bred livestock, their ability to fight any kind of infection has actually gone down because of the fact that they have been bred for the other properties. As a result, in order to keep them healthy and alive, most of the time the livestock breeders, not the breeders, the people who are farming, dairy farming or wool farming, etc., they end up injecting their animals with enormous amount of antibiotics. And unlike the human scenario where the use of antibiotics is somewhat controlled, in the context of livestock, the use of antibiotics is hardly controlled. As a result, most of these people, they end up pumping antibiotics at a high rate into their animals, and most of these antibiotics, they actually do not get absorbed in the body. Most of it actually passes out through urine or fecal matters and ending in the environment. As a result, the environmental load of antibiotic goes up, and obviously that creates a selection pressure even for those microbes which are otherwise not pathogenic, which are otherwise not even inside the body of the sheep or the cow. Now, because of this stuff, this is like one of the biggest reasons for which there has been a great increase. The second, or rather the third thing that also happens is the same stuff in the context of agriculture or aquaculture. So, nowadays lots of people, they end up spreading antibiotics along with pesticides for their crops, and of course people who are into aquaculture, fish culture or prawn culture, etc., those people also end up using tremendous amount of antibiotics. And finally, there is a lot of residue that happens from pharmaceutical manufacturing, and this also gets into the environment. And of course, when it comes to antibiotics, improper disposal of hospital wastes happens to be one of the biggest culprits. Now, all these things, they taken together, they are all of them are happening simultaneously, right. So, taken together, these have increased the baseline level of antibiotics in the environment to an epic proportion. However, they are not the only culprits. I mean, these five are probably the biggest culprits, but there are other things that also end up increasing the selection pressure on the bacteria. For example, as I told you that bacteria has these efflux pumps which are active not only to throw out antibiotics, but also to throw out other heavy metals and chemicals and so on. Now, because of all kinds of manufacturing processes, the concentration of heavy metals and these various kinds of industrial chemicals, pollutants, these have greatly increased in the soil and atmosphere and so on. Now, because of these, there is not a direct selection for antibiotic resistance, but there is an indirect selection for antibiotic resistance. So, whenever a bacteria is encountering these chemicals, it is actually ending up, among other things, increasing its efflux activity, and that as a secondary for a reason can end up increasing its antibiotic resistance. The other very interesting thing that we found out was related to fluctuating environment. So, this was an experiment that we did in our lab, wherein we took environments, pH and salt and so on, and we just fluctuated the environment, we varied them unpredictably. So, the whole idea was that in natural world bacteria is always going to get environment which is continuously changing and most often unpredictably. So, how exactly they end up combating that? And what we found was that when we changed this environment unpredictably, then the bacteria, the first thing that they did was they increased their efflux activity, and because they increased their efflux activity, as a correlated response they ended up getting increased antibiotic resistance. Now, think about it, these were bacteria which were never exposed to any antibiotic, yet because the efflux activity had gone up, they had become more resistant to antibiotics. So, why is this critical? This is critical because if you look at what climate change is doing, it is not only warming up places, it is not only changing the pattern of precipitation, it is also leading to a lot of increase in the variability in various parameters, environmental parameters, and our work is showing that that increase in the variability alone under certain circumstances can end up increasing the bacteria's ability to combat antibiotics and other, in the context of bacteria it is antibiotics which matter. Okay. So, as a result of all these things happening together, there has been a massive increase in the selection pressure for antibiotic resistance on bacteria, not only the pathogenic ones, on all of them. And this in the context of bacteria is a huge thing. Why? Because there are certain properties of bacteria which makes them extremely potent against these kind of selection forces. What are those? Number one, remember that bacteria have massive population sizes, right, and they also have extremely low generation time, right. They are multiplying very fast, they can multiply very fast. Now, because of this there are certain advantages a bacterial population has, which for example a human population or a goat population will not have. Like what? Because the population size is very high, so that means they are dividing continuously, they have a very high chance of hitting beneficial mutations. Remember, mutations by themselves will arise randomly with respect to fitness, right. Most of the mutations that will actually have a change in terms of the fitness, most of them will end up being deleterious. But in spite of that, even if there is, let us say, a 1 in a million chance or a 1 in a billion chance of hitting a beneficial mutation, that kind of a mutation is much more likely to occur in a population which has large size and is replicating extremely fast compared to, let us say, a population like ours, which although the size is large, but we are replicating very slow. So, the chances of getting the beneficial mutation is high, and more importantly, because the population size is so high, remember we talked about the selection drift balance. We know that as the population size increases, the role of drift goes down and the role of selection goes up, which essentially means that in bacteria for high population sizes, selection is most probably going to be a very dominant force. And as we have seen, selection anyway is a very strong force of evolution, and it can lead to extremely high levels of change, high magnitude of change rather fast. So, if you have a scenario where selection is operating extremely well, that is a recipe for disaster. To make life more interesting or scary, whatever you call it, we are also having a lot of zoonotic transfers. What does it mean? So, zoonosis essentially means the situation wherein bacteria and other microbes, they end up moving from their regular hosts to other organisms. So, in this particular case, we are talking about some bacteria moving from some organism to humans. Now, because of various reasons, there is a lot of increase in zoonotic transfers. So, for example, the biggest example that I can straight away think of, although not in the context of bacteria, is viruses, right. Zoonotic transfer from bats, we all know where it went. And not only do we have transfers from wild animals, there is a lot of zoonotic transfer which is happening from domestic animals, and that is a lot more scary, because as I told you that domestic animals are being continuously pumped with a lot of antibiotics. So, many of these pathogens which normally are not human pathogens, but let us say they are in the domestic animals, they are getting lots of antibiotics. So, they are becoming resistant, and at some point due to whatever mutation, they end up shifting from those animals to us. So, for example, if you remember the bird flu, right, the swine flu, these are all examples of zoonotic transfers which are happening from other domestic animals to humans. To make life even more surreal, there is climate change also plays a role over here, because many a times you end up getting stuff from permafrost. So, in 2016 there was a very major anthrax outbreak in Russia, and they could not really figure out why it was happening till they figured out that due to global warming, the permafrost. So, okay, what is the permafrost? Permafrost is basically that part of the soil or the sea bed which is permanently frozen. The temperature is permanently or for a very long time under 0 degrees Celsius. Now, because of global warming, the temperature in many places in Siberia and other places with permafrost, the temperature is increasing. Now, because that is increasing, the permafrost is melting. Now, the permafrost actually ends up trapping lots of microbes inside it. More importantly, in places like Siberia, it traps the carcasses of animals which have died long back, but got frozen before they were decomposed. So, in this particular case, it turns out that some of those reindeer carcasses in Siberia, they were actually having very deadly anthrax germs, and they had gotten frozen. And then this happened long back, hundreds of years probably back. But when the permafrost got frozen, then all these carcasses got exposed, and then all the anthrax came out, and that anthrax was responsible for the 2016 outbreak. Absolutely scary kind of a scenario. Now, obviously, if you have all these bacteria coming in, these are all new organisms, okay, and therefore sometimes these new organisms can end up making life very difficult, because they might have genes which through horizontal gene transfer can be transferred to the existing pathogens, making them even more deadly. In the context of antimicrobial resistance, that is actually our lesser worry. Our bigger worry is that because AGT is extremely common in bacteria, suppose there are so many species of bacteria out there, right? Environmental load has increased. Suppose there is a non-pathogenic species of bacteria that has ended up getting the mutation which makes the bacteria resistant to some antibiotics, then from that non-pathogenic strain, that thing can end up transferring into the pathogenic strain, which then can become really problematic for us. And the issue is that we know that it is happening, and it is happening at an extremely alarming rate. And the whole thing has been complicated even more because we human beings, we obviously have all the countries are connected to each other by commerce and travel. So, suppose let us say I am sitting here in Pune and I am let us say doing some farming, and in that farm I end up putting in some antibiotics, because of which some bacteria ends up developing some resistance, and let us say I whatever I create some crops and I do not wash my crops properly, and that crop gets sent over to wherever, say Africa or China or US, wherever, and there that bacteria ends up transferring that gene to another bacteria in that country, and therefore those guys over there who never ever were exposed to that antibiotic, they did not even use that antibiotic, suddenly they will find that people are not responding to antibiotic treatment in their country simply because the resistance evolved in my country and went over there through commerce. So, this kind of a scenario has made the entire thing a living nightmare, and to make it even more complicated, the one major thing that we are seeing nowadays is what is known as multiple drug resistance. What is that? So, when you have simultaneous resistance, also called cross resistance to multiple drugs. Now, why should such a thing arise? There are many ways in which it can arise, some of which I am quickly telling you. So, one of them is, suppose you have a bacteria, but because typically you will get just one or two mutations happening in that, but suppose there is prolonged exposure to antibiotics, as what will happen if the load in environment is high. Because of that, the same bacteria might end up getting multiple mutations in multiple genes, or the same lineage can end up getting multiple mutations in multiple genes, which can end up making it resistant to multiple drugs at the same time. Or, as I told you, there is horizontal gene transfer. Now, when there is horizontal gene transfer, remember, one of the things that bacteria transfer are plasmids. Now, plasmids are circular DNA molecules, and they can contain multiple resistant genes at the same time. So, one plasmid can contain multiple resistant genes. So, in one shot you can have a scenario where resistance to 3-4 genes are going to be transferred straight away, okay? And obviously that makes life very difficult. And the third thing that is happening, and this is what is making life even more crazy, is the evolution of so called supergenes. So, for example, there are this group of enzymes known as ESBLs, extended spectrum beta lactamases. So, beta lactamase, this is a kind of an enzyme which ends up breaking down certain antibiotics, but these particular group of enzymes, ESBLs, they end up allowing the bacteria to cut out many kinds of antibiotics at the same time, and because of this it makes the pathogen resistant to a wide class of antibiotics, including very recent 4th generation antibiotics, and this makes this kind of resistance extremely hard to treat. And right now ESBLs have spread into E. coli, Pseudomonas aeruginosa, I think also staph, and many other extremely hard to treat organisms. So, what does one do? How do you combat this? Obviously, the best thing to do is more rational prescription of antibiotics and avoidance of self-medication. Now, the problem over here is, this is something that is doable, but due to social reasons, as I discussed, it is hard to do this. More critically, see, this is a community thing. As a person, I can say that, look, I will not take antibiotics, okay, I will let the infection run its course, even that if that means that I will have to be on my bed for 10 days. The problem is, suppose my neighbor does not think that way, okay, my neighbor ends up taking the antibiotic anyway, and therefore the resistance develops in that neighbor, but remember, this is a community thing. So, the resistance that develops in that neighbor, it does not stay inside the neighbor, it passes on to the environment, and therefore even if I am not using antibiotics, I am not immune from getting bugs which have got their resistance from somewhere else. Therefore, many people end up thinking that why bother, until and unless the entire society signs up and very religiously does not use antibiotics, I am anyway going to get antibiotic resistant bugs. So, why should I go through the pain of fighting the disease? Let me also have the antibiotics. So, which is why, although it is very easy to say, it is very difficult to implement that first step. As I said, huge amount of antibiotics are used in agriculture, animal husbandry and aquaculture. So, somehow one needs to come up with good quality policies to monitor and rationalize the usage of those antibiotics. One thing that we have noted is that many a times when bacteria end up developing resistance to one kind of antibiotics, they become sensitive to another kind of antibiotics. This is what is known as collateral sensitivity. So, one needs to monitor what mutation or what resistance is associated with what sensitivity, and then if possible one can give drug combinations in such a way such that you let us say resistance to antibiotic A makes you sensitive to antibiotic B. So, give a combination of A and B so that every time a bacteria tries to become sensitive to A, it ends up being killed by B. Now, of course, this is not very easy to do because these patterns of collateral sensitivity themselves are very labile. They end up depending on all kinds of epistatic interactions in the genome, but if one can take care of it, this is one way in which at least to some level the spread of resistance can be stopped. Of course, the best thing is prevent the infections, improve sanitation levels, as far as practicable vaccinate at least against the most common diseases. So, the whole idea over here is that try to make sure that you do not even need to take antibiotics. That is what is going to reduce the overall antibiotic usage. Easier said than done, of course. And finally, we have to step up our rate of discovering new drugs. Right now there has not been any new class of antibiotics for the last several years. So, in 2025 finally they announced a new class of antibiotics after a gap of 30 years. Okay. This is partly because antibiotics are very powerful. So, most of the time, although there are many candidates, when it comes to trials, human trials, many of these candidates are thrown out simply because they end up having extremely high side effects, which is why developing a new antibiotic is not easy, but somehow or the other we have to do something to get more drugs. So, that was about how evolution of microbes can create problems for us, but turns out that under many situations our own evolutionary history can affect how we end up getting diseased conditions, and that understanding and the corresponding medical practices, they form a very fascinating subdiscipline of medicine known as evolutionary medicine, but that is what we are going to deal with in our next discussion. See you then. Bye.